Welcome to Lecture 1, The Eye. Before we begin about the eye, let's consider a question. What would happen if you used more than 10% of your brain at one time? This question is posed in a movie that appeared last year called Lucy.
It is estimated most human beings only use 10% of the brain's capacity. Imagine if we could access 100%. Interesting things begin to happen. Yes?
Professor Norman, my name's Lucy. I just read all your research on the human brain. It's a little rudimentary, but you're on the right track.
Thank you.
I have access to 28% of my cerebral capacity. I can feel every living thing.
Since when did you start writing Chinese?
Since an hour ago.
What happens when she reaches 100%?
I have no idea.
So, one of the things that you'll discover in this course is that we like problems. And to solve problems, we pose a few questions. So what do you think? What would happen if you could fire more than 10% of your neurons at the same time? Would you A, become like Lucy? Would you simply become tired? Or C, would you faint? Well, the answer to that question is what we'll study in this course. And we'll find out, for example, that the firing of action potentials takes a lot of energy. If we activate more than 1 or 2% of our neurons at the same time, that's 1 to 2 billion neurons, we do risk fainting because we have depleted all the oxygen that the heart can supply to the brain. Now the brain has developed so it's very efficient. iMac uses about 150 watts of energy. Your brain consumes only 20. So how the brain does that is what we'll cover in this course. How does it compute all that it must with so few neurons? Before we do that, we have to come to appreciate how some of the building blocks of the brain work. And we'll begin today with the eye. The eye is in fact something that evolved from the brain. In the fetus stage, a bulge grows out of the growing brain and becomes the eyeball. So when we look at the neurons of the eye, we are looking at the neurons of your cortex as well. So this is a picture of the eye. And you can see light coming through the lenses bending the light rays onto the back of the eye, which is the retina. This is where all the neurons lie that convert light energy, photons, into electrical energy. And that electrical energy is then sent down as action potentials down the optic nerve to the rest of your cortex. Now, the lenses are as follows. First, you have on the very outside a fixed lens, one of constant shape. Inside, behind this lens, you have a flexible lens. The flexible lens is attached via springs to your ciliary muscles. These muscles encircle the lens like a donut and can contract and relax. Here we see what happens when it contracts and relaxes. When it contracts, the diameter of this donut, this is just a slice through it, we'll see in a moment how the whole thing looks like, gets less. As a consequence, the tension on these springs become less, and the lens springs back to its normal round shape. Okay? When the muscles relax, the diameter becomes larger, and this pulls the lens into a flat shape. So this process allows us to focus the image of things at various distances from the eye, from far to near, onto the back of the eye. We can see here again the lens becoming flatter, rounder, flatter, rounder, and because of the contraction of this ciliary muscle, the muscle that pulls on these springs. So the flat lens, round lens, flat lens, round lens. When the lens becomes more round, we can focus near things on the back of the eye. When the lens becomes more flat, we can focus far things on the back of the eye. Now, what happens when the lens isn't, when things don't focus, or why is it that sometimes you need glasses? The shape, the ability to focus the image on the back of the eye depends on two factors. First of all, the shape of the eyeball. It can be too short or too long, or it could be the shape of the lens. The lens can be too flat or too round. Either of this can change the focus on the back of the eye. Okay? And you can see that the image becomes sharpest when the image is focused directly on the back of the eye. Now, either of these problems, too long or too round, causes something called nearsightedness. You can focus on things that are near, but you can't focus on things that are distant or far. And to fix that problem, you need concave lenses, glasses of this shape. They're narrower in the middle of the lens and thicker on the edges. If you have either of these problems, the eye being too short or too flat, you need something called the convex lens. You are farsighted, therefore able to look at things far away, but can't focus on things near without these convex lenses. And this lens is thickest in the middle and thin at the edges. Now, as you get old, the lens becomes stiff. So this is a normal lens. You can see every time the muscles contract, it becomes nice and round. This is an old lens. Every time you contract here, the lens gets a little more round, but not as much as it should. As a consequence, it's not round enough to allow you to focus on things that are near, and you tend to move things farther away in order to bring them into focus. Okay, so another problem. What type of lens do you think you'd prescribe if you had this problem? You couldn't focus on things that were near. Would it be no lens? Nope, that won't help anything. Will it be a concave lens? No, you can see here the problem becomes worse. The newspaper reading becomes more flat. Okay, and the object, the focus, occurs even further behind the eye. If you, on the other hand, use a convex lens and a convex lens of the right roundness, you can get the image focus back onto the retina. Okay? And you can see the newspaper clearly. Okay, the other part of the eye, in fact, the thing that is most visible to the eye, is the iris. It's what gives the eye its color. You have blue eyes, or you have brown eyes, or a variety of shades. And this iris, inside this iris, you have this black spot. That's where light enters the eye, and that can become more narrow. When it becomes more narrow, less light enters the eye, and makes light-sensitive receptors less able to become saturated with too much light. So it protects these rods and cones, the receptors, from too much light. Allowing you to read, to see clearly, even in bright sunlight. Now, the other byproduct of the iris is that, as you can see here, it makes the pupil narrower. And when it does that, if the image isn't focused perfectly on the back of the eye, instead it's focused back here, there's a certain blur on the retina itself. Okay? When you close your iris, that makes this blur smaller. Okay? And the same thing happens in a camera. When you reduce the aperture, you allow the camera to have a greater depth of focus. Okay, like the eyeball here. So, what happens on the back of the eye? What do you have? Well, you have 5 cell types. You have cones whose ends here are shaped like a cone, and hence the name. They're one receptor that is sensitive to light and converts the light energy into electrical energy. The second, and the cones are very important for seeing various colors. You also have things called rods, and all the rods are sensitive to the same color, which you interpret as either gray, black or white, or various shades of gray. The next cell type that's important is the ganglion cell. That's the output cell. All the electrical activity from the eye passes through these cells, and that the axon of these cells is what sends information to the cortex. Next, we have bipolar cells. They are what connects the receptors out here to the ganglion cells down here. In addition, we have horizontal cells, which combine information from several receptors. They can be cones or rods, both types. And they determine how much of the retina each bipolar cell sees, and in turn, how much of the retina each ganglion cell sees. In addition, and finally, we have amacrine cells, and they do the same thing as horizontal cells, but mostly for the more peripheral rods. Okay, what happens when light strikes these receptors? The odd thing about what happens is that when it turns dark, you can see here, dark, potential rises. Light, potential decreases. Dark, potential rises, decreases. So it looks as if these receptors respond to dark. They like dark as a stimulus. In addition, the interesting thing is that in terms of what type of potential, you have potential that rises slowly in some cells. And that occurs in all the cells except for these ones down here. So here we have not action potential, but graded potentials. In the other cells, the ganglion cells and the amacrine cells, one has action potentials. Not all amacrine cells, but only some. Now, why is it? Why is it that some use one and some use the other? Well, the answer is that action potentials are not very good at telling you exactly when things change. Okay? To determine when things change, the cortex has to wait a certain time. For example, here you have the ganglion cell firing at a certain frequency. Okay? It fires at 10 hertz, 10 times a second, every 0.1 seconds. So each of these action potentials are 0.1 seconds apart. So in order to determine whether or not these action potentials are slowing down, you have to wait at least 0.1 seconds. You can see here that they're firing 0.1 seconds, but now you have to wait until the next action potentials to determine that it's faster or slower. So graded potentials have the advantage in that they can allow continuous and rapid information. Okay? The unfortunate thing is that information cannot pass through from one neuron to another neuron unless they're close together. But in fact, that's what occurs for most of these neurons in the retina. For the receptors, for the bipolar cells, and for the horizontal cells. It's just the ganglion cells and some amacrine cells that have to resort to the less secure action potentials. And they do so because they, for example, the ganglion cell needs to transmit information all the way to the cortex. Graded potentials will not travel that far. So it's forced into using action potentials. Okay, next question to ask is, why is reading difficult in low illuminations? I mean, it seems natural that it's almost impossible to see, let alone read this newspaper in a dark environment. Well, the answer to this question has several parts. First part is that the retina is not uniform. This middle part, right here, is called the fovea. And this is where the eye is pointed when it's interested in certain features. So the eye turns and points the fovea at whatever it is that you're interested in. Here one finds only cones. In the periphery, one finds primarily rods. Okay, so mostly cones in the middle, mostly rods, well, only cones in the middle, and mostly rods in the periphery. The second answer to this question is that cones are less sensitive to light. Okay, so if you're in a dark environment, these ones here, the rods are more sensitive, and you see things better. You might have noticed that when you look out in the dark sky, that you might spot a bright star sometimes. But when you look at it, that is, point your fovea towards the light star, it disappears because the cones here are less sensitive to light. The third part of the answer is acuity. The periphery over here has less acuity. The fovea here has high acuity. When I tell you to look at this diamond, you point your fovea at this diamond, and your periphery sees, if you don't take your eyes off the diamond, the other letters, and you'll notice that it's hard to see clearly what's happening over here without taking your eyes off that point. Why is that so? Well, there's several reasons for that. One is that in your periphery, your receptors become large, and as a consequence are spaced farther apart. They have a lower density. Also, these receptors have a large convergence. The horizontal cells and some of these amacrine cells are sending information from lots of these receptors to the ganglion cell. As a result of this large spacing and large convergence, you have low acuity. In contrast, in the fovea, the receptors are small. Your cones are all small in shape in the fovea, and only a few of them converge onto a ganglion cell. The result of the small spacing and low convergence gives you high acuity. That's what you need when you read. In the retina, there isn't just small and large, but a continuum of ganglion cell sizes, from small to larger still to the largest. So by daytime, you actually only your very center of the eye sees in detail and in color. It might be most often this is a surprise to people that only this small part of your retina can actually see in detail and in color. On a dark night, only periphery sees, and only in black and white, and only with poor resolution. The fovea itself is blind. Now, the optic nerve is small compared to the large retina. So what happens is that things get compressed. Okay. Most of the optic nerve is sending information from the fovea. Okay. Half of what you see, half of the fibers in here are from ganglion cells that are coming from the fovea. Now the fovea, in fact, sees just a tiny percentage of the whole retina. So 100% of the whole retina would be this distance, this distance. The fovea itself only sees 1% of that. That's about twice the width of your thumbnail when you hold it at arm's length. Compare that. Put out your thumb, look at it, and compare twice its thumb width to what the rest of everything that you retina can see. It's quite a difference. And yet half of that is taken up by fibers heading from the fovea. The receptive field is what I want to cover next. The receptive field is a very important complex, but can be difficult to understand at first. When we talk receptive field, we talk about neurons. And for example, here let's say we have to talk about the receptive field of the ganglion cell. And the definition of this receptive field is the area of the retina over which light stimulus changes the activity of this particular ganglion cell. So which of these receptors, rods or cones, are connected to this ganglion cell? Now in this case, you can see this is the ganglion cell firing at the action potentials of this rate. Which of these receptors are connected to it? Well, according to the definition, we simply explore each of these receptors by shining light at it. If we move it over to this receptor, nothing changes. Okay? So this is not inside the receptive field of the cell. If we move it to this cell, the firing frequency goes down. So that's a change, and so that receptor is part of the receptive field. Again, the frequency here goes up, so again that's a change, and therefore this receptor is part of the receptive field, as is this cell. And finally, this cell, the activity gets back to normal, so that what we found in the beginning, so that cell is not part of the receptor field. So only these central three receptors form the receptive field of this ganglion cell. So this is important because this definition will be used for many other neurons in the cortex. That applies to other visual neurons. We'll talk about neurons in the cortex and the shape of their receptive field, and we'll study it exactly the same way as we looked at this ganglion cell. Or we'll look at other sensory modalities such as touch. So the same definition applies, except that the retina is replaced by the word skin, and touch pressure replaces the word light. Okay, in the retina, we have many types of ganglion cells. Two main types are on-center, off-surround. So this is an example, on-center. So all the receptors in the center produce an increase in firing rate of the ganglion cell, and all the receptors over here produce a decrease in activity of the ganglion cell. And this cell tells you that the center is lighter than the surround. In contrast, one would have receptors called off-center, on-surround, and in this case, all the receptors in the center produce a decrease in activity in the ganglion cell, and in contrast, the receptors in the surround produce an increase in activity in the ganglion cell. As a consequence, this receptor responds best when the center is darker than the surround. Let's suppose somewhere over here we had a ganglion cell that produced these action potentials. How would we determine what the shape of its receptive field was? Before we do that, let me explain that what you see here is a picture of the retina. These dark spots are the arteries and veins of the eye, would send blood flow through the eye, and they course meet here in the middle, and this white spot here is the optic nerve. So they exit at the optic nerve. Over here, you can see a little darker spot, and you can see that that is where your fovea is. So moving light all over these areas doesn't seem to affect this ganglion cell that we're recording from. But when we move a little bit over here, we find, just off the fovea, we find that the action potentials become more frequent. So you do have neurons here, receptors, within the receptive field. If we go further over, we find that the activity drops below what we saw for the out here. Okay, so this is higher here and then even lower here. So this again is within the receptive field of the ganglion cell, and then we move out further, we get back to higher and then back to baseline. Okay, and if we track this around and draw it out carefully, we'll discover that we have this type of receptor field. We saw here close to the fovea an increase in activity, then a decrease, then an increase, and back to baseline. And if we plot it out carefully, we'd get this shape. So that, by the way, is an off-center, on surround. Okay, the other type of ganglion cell is something called a rapidly adapting or phasic ganglion cell. We saw, the neurons we saw up now either had an increase, then stayed high, or a decrease and stayed low. Here we have something in between. So when we turn the light on, you can see the ganglion cell starts firing more, but then gradually comes back to the rate at which it was firing when we first encountered it. When we turn the light off, it silences briefly and then gradually comes back again to the same rate as we saw initially. So this just has a phasic response, just a short response when we turn things on or turn things off. So that is very good for responding to things like a light that turns on or turns off. Now, how does these neurons connect to produce, let's say, an on-center, off-surround? Let's suppose this was a ganglion cell connected as you see here. What happens, and it has a center and a surround. So this receptor is in the center, this receptor is in the surround. This receptor is an on-center. Okay, so we expect this light in the center to produce an increase in action potentials in this ganglion cell. So let's see what happens here. When light goes on, the voltage drops. Less transmitter is released. The voltage here rises. When the voltage here rises, then the more transmitter is released here, and the action potential rate goes up. Now, when light goes down, the potential rises here because the transmitter here is an inhibitory transmitter, okay? So when you go from high, which is lots of inhibition, to low, which is little inhibition, we start becomes less inhibited. As a consequence, the voltage rises. A simple way I like of thinking about it is that whenever I see an inhibitory synapse, it's like a negative side. That negative side flips whatever happens here. So whatever happens here, the opposite happens here on the other side, the negative side. So this is falling, then this is rising. When it's a positive or excitatory receptor, the same thing happens from this to this. The same response occurs. So what happens in the surround? When it happens in the surround, we have this neuron connected to the horizontal cell, which connects to this receptor, which then sends information to this bipolar cell. The other piece of information is that this horizontal cell has an inhibitory influence on this receptor. So when light occurs here, the potential here goes down, this goes down, this goes up, this goes down, and this goes up. Okay. Again, we can look at it in terms of plus and minus. This is a positive receptor, so whatever happens here as well. This is an inhibitory connection, so the opposite of what happens here happens here. So if this goes down, this will go up. Again, this is an inhibitory connection, so if this goes up, this must go down, and this must go down as well. So this light in the periphery produces a decrease in action potentials in the ganglion cell, and as a consequence we have an on-center, off-surround ganglion cell. Next topic, okay? What's the function of all this? You've got this complicated circuitry. Why have it? Well, the main function of this ganglion cell, of this type of response, is that it accentuates edges. Now, artists like Seurat discovered this many, many decades and centuries ago. For example, here you can see that Seurat found that if he made this darker than here and this darker than here, he would have a more sharp edge created. As a consequence, emphasize this part of the painting. Well, what happens? How does this happen? How does ganglion cells accentuate edges? Well, you can imagine, here we have a ganglion cell way out here, another ganglion cell way out here. Both are not changing in their activity. There you can think of them as being asleep, okay? The activation from the center cancels the inhibition from the surround. Here is light, and here is dark. On the dark side of this edge, okay, the same thing happens. Here, everything excited from the center gets canceled by everything that gets inhibited by the surround, giving you the same sleepy activity. So you might have little or no action potentials produced by these neurons, and most of these neurons that are far from the edge. The only ones that will awaken is those at the edge, those that are receiving information from this edge. And this on-center receptor will tell you that here we have a lighter portion, because the amount of excitation will exceed the amount of inhibition, or missing this part. This one here will say, okay, this side of the edge is darker, because again, the amount of inhibition will be exceeded in terms of excitation. Not as much excitation will occur. Only this part will be excited. So, we asked this question: how does the brain compute what it must with so few active neurons? Well, one of the ways is by only computing change. Okay? We keep the numbers of active neurons to a minimum. In the case of the retina, the only active neurons, are the ones at the edge. So what we see is an accentuation of edges. Now, if you look at this, what is actually there is a gray, then a darker gray, then a darker gray still, then a darker gray still. But what we perceive is this, because of our ganglion cells. We see it's lighter over here and darker over here. Okay? The edge is becoming accentuated. Again, the same thing happens here and here. Okay? And so this illusion is the consequence of our ganglion cells attempting to accentuate all our edges. What's another property of these ganglion cells? Okay, they're measuring the difference across an edge. Okay, so when they measure the difference across an edge, the difference over here to over here will be a certain difference, and the difference from over here to over here will be a certain difference. Imagine that these are letters. This is just one letter on a page that you view in a darkened room, like a lecture room with lights down. Imagine this is the same piece of paper with the same letter, now viewed outside, outdoors, by a bright sun, under bright sunlight. The change between this, from here to here, is the same as the change from here to here, because it's the same piece of paper. Okay, this is just as dark, and this is just as light. So you want the brain to reproduce this same change. Okay, now, and it does so quite faithfully. That's why you have constancy. But it does so is somewhat surprising, because if you look at the sheet of paper, okay, the absolute, not the change, the amount of light coming from this situation and this situation, and then compare, let's say, the H to the paper, you'll find that the paper here, the letter H, the black letter, is lighter than the white page. Okay? The white page, under low illumination, reflects less light than the black letter seen outdoors. Okay? So there's a huge difference as to how much overall light is transmitted in this situation and this situation. But by measuring just the difference between the center and the surround, which is what ganglion cells do, they produce constancy. Black appears as black and white appears as white. Okay, the last topic I'd like to cover is color. What is the advantage of color? What do you think is tastes better? These fruit bowl seen with color or without color? Well, you can see that it's very hard to tell whether the fruit is ripe when it appears in black and white. And that's thought to be one of the main reasons that pushed evolution towards developing the ability to see color. How do you see color? Well, the starting point for seeing color is the fact that you have different types of cone receptors. So this is the response of a cone receptor to different light frequencies. This is the blues, this is the reds, yellows and greens in the middle. So this receptor responds best to green light. Okay. Now, what's the difference? Let's suppose that's all the receptors that you had. What's the advantage of having more receptors? Well, suppose you had this situation. This was your green cone, and you viewed this field here. The background, that's this color here, and this color here on the chart, would produce this response. The foreground would be this color, and this color here produces this response. That response is the same as this response. As a consequence, what you would see if you only had green cones is this, nothing. Both the center and the surround give you the same response. Not very good survival value. It's also not very good for detecting things like whether see a ripe apple in a green tree. So what happens when you have two receptors? We have add a red cone here. Well, in this case, the background is clearly visible. The foreground is clearly visible from the background because B here produces this response, a very good response, whereas A here produces very little response. So most species have developed at least two types of cones. We have developed three, and a few species have in fact developed more than three to do even better. Now, we need different types of cones in order to perceive color. But we can't rely on just the cone response itself to see color. Why is that? Well, if you look at these curves, okay, either of these curves, they respond best to this color, but they also respond pretty well to the other colors as well. Okay, so anything, from about here to here gives you a fairly hefty response. And if you're looking for here, for example, and the response gets larger, is it because the color is changing, or is it because it's becoming lighter? In other words, this whole curve is shifting up. Well, we'll find out in the next in a short time that to do that we have a special type of cell, not the ganglion cell, but a cell that occurs in our visual cortex that we'll look at in a moment. Okay? Well, let's again just briefly review what we do have. We have three types of cones: red, green, and blue. And these are the wavelengths to which they respond. The next important thing that you should take from this is that mixing different colors, so we can turn them on, turn these receptors on, not just one at a time, but all three at a time. When we turn on two at a time, we see yellow. Red and green combine as yellow. Now this is different from mixing paint. Okay? Combining light is not the same thing as mixing paint. If we turn on all three receptors, we see white. Okay? And certainly that's not what happens when you mix these three types of paint together. We see something very far from white. Okay, if we look at the very center of the fovea, let's go back here, this very center, and then this is expanded. Okay. We see that we have these cones here in the very center. And these cones are interesting because they tend to form in groups, okay? You have a bunch of red ones together, here a bunch of green ones. The proportions of these three differ from person to person. And the numbers of blue cones is usually the least, and the numbers of red cones is usually the most. In the very center again of the fovea, so before we looked at just within the fovea, but now we're looking at the very, very center of the fovea, we find that blue is missing. We just have red and green. In contrast, in the periphery, we find that we have, in addition to a few big cones, we have big rods. Okay, so the size gets bigger of each receptor, so the density gets smaller, and we find that the size of the cones has become bigger than that of even the rods. Color blindness. If you look at this, when it's gray, you see nothing in color, and you have no color blindness, you should see the letter 10. Over here you should see a 0. Over here you should see a 1. If you don't see a 0 and a 1, you may have color blindness. Most likely you don't, because this may, the colors reproduced by your terminal may not be exactly the right colors to detect these differences, okay? So you need to check this out with somebody that has a screen that's perfectly tuned to the correct colors. You can have various types of color blindness. You can be missing just one type of cone, either no red cone, no green cone, or no blue cone. Or you can be missing two types of cones, or you can be missing all the cones and just, or you can have the shape of this be different from normal. So the shape of the response to these wavelengths for the red cone are not the same shape as normal shape. Okay, let's then give you a problem to solve. Suppose you lost all three types of cones. As a result of this, you would not, A, be able to see a red apple, be able to see in dim light, or be able to read. Which of these would occur? Well, the answer is read. Okay. You can still see with the rods, okay? You can see shapes of things, you just can't tell their color. The rods, it will still respond to red light, okay? As I said, you can't tell it's the redness of it, but you can still see the red apple. You might have more trouble distinguishing it from the green leaves, but you can still see a red apple. And you can still see in dim light because, as we saw, cone rods are quite good to seeing in dark environments. What you'd be missing if you got rid of all these three is all the receptors in your fovea. And as a consequence, you'd have a poor, you just see with your peripheral retina and have a coarse acuity, which of course is not good for reading. So we only have three cones, but we can see such rich colors in the environment. How do we do that? Well, first of all, if you look at how many hues you can distinguish, if you take all wavelengths of light that the eye can respond to and lay them out in this circle, we find that we can detect 200 shades of light. If we put them in the circle, then complementary colors are colors that are on the opposite side of this circle. If we look at how we can mix different colors, we find that we have something like 200 levels of saturation. That is, 220 levels of saturation. So, for example, if we mix a green here over here with this purple over here, it will change we're going to detect something like 20 different differences of combinations. The interesting thing again, as we pass through the center, we combine these two colors in equal amounts and we see white or gray. If we combine colors over here, we can combine red and green over here. We'll see a yellow, something between, but these are not complementary colors. Okay, if next we make them lighter or darker, we can see these circles get smaller. That means we can distinguish less differences in color in very bright environments, and less differences in color in very dark environments. But we can still detect something like 500 different levels of brightness. So if we take these numbers and multiply them together, we find that we can detect 2 million different gradations of color. So this is why paintings can be so filled with different hues and colors that they can have such a profound effect on the eye. Okay, what happens here? Try keeping your eye at this point here. Okay, if you do that and keep in mind what happened in the periphery, but don't look at it, okay? Just keep your eye on the center. Gradually you should see that a dark blue light starts to appear, moving round and round and round on these yellow lights, okay? And if you look at it, keep looking at it even further, okay? Keep concentrating further. Eventually, the yellow lights disappear totally, and all you're left with is the blue light going round and round. It's very important to maintain your fixation and not get distracted by the blue light, okay? It's easy to just move briefly to the blue light and ruin everything again, and having to adapt to the color. Okay. So, again, what you should have seen is gradually the yellow lights are replaced by a blue light moves round, round, round. Why is that? Well, again, I like you to think about it. There are three possible choices here of what's happening. There is a rotating blue dot that appears late in the scene. Okay? The yellow dot dims late in the scene. Okay? Late in the scene, the yellow dot starts becoming dim. And lastly, the yellow dots are briefly turned off in sequence. Okay? So they're one after another. And actually, there's no blue dot ever appearing on the scene. But you see it. So pick your choice. In the meantime, let's look at this. What do you see here? If you look carefully, just keep your eye constantly over here. You should see, with time, color appearing. Okay? Gradually, instead of when things go gray, instead of turning gray, this starts becoming reddish over here. And other colors appear as well. This reddish becomes greenish. Okay. Why is that happening? Again, it's occurring because cones adapt. Do the same thing here. Look at the X. If you carefully keep your eye on the X, but concentrate on what's happening to the white when the lights go off. Without taking your eye off the X, it's a hard thing to do, but you'll be rewarded with what you see. Gradually, instead of white, colors will start appearing in your periphery, and they'll be different from the colors that are shown initially. Instead of green, you should see a purple. Instead of purple, you should see a green. Instead of red, you should see a cyanide or pale. And instead of a yellow, you should see a blue. So what's happening here, the opposite of this appears here. The opposite of this appears here. The opposite of this appears here. Okay. Here, you see its complement, which is over here. Here you see its complement again, which is over here. Here you see its complement as well, which is over here. So what you see is complementary lights appearing as afterimages. So it appears that light is somehow treated in push-pull pairs, okay? And that, for example, yellow and blue are push-pull pairs, as are green and magenta, as are red and cyanide, okay? So if red is shown for a long time, then cyanide will appear as its afterimage. If yellow is shown for a long time, then blue will appear as its afterimage. So we'll see how these push-pull pairs are formed from ganglion cells next. So let's briefly return to our problem. Okay. We have these three choices of what's happening here. Which is the correct answer? Yes. What in fact is happening here is that the yellow lights are turned off in sequence, okay? Briefly, this yellow light is turned off, then this one, and so forth, right around. And what appears in its place is the after image of a yellow light, which is blue. So when it gets turned off, a blue after image appears in this location briefly. How does that happen? That happens because of special cells located in your cortex, and these special cells are the ones that are sensitive to color. These cells are called double opponent cells. They're called double opponent cells because whatever you have in your center has an opponent in the surround, and whatever you have in the center also has an opponent in the center. So you have opponents in the center and opponents in the surround. So here you have gets excited by blue and inhibited by red. Here blue and inhibited by green. The opposite happens in the surround. So this is an example of a yellow-blue opponent cell. Combination of red and green is yellow, and its opponent is blue. What happens when you view, then, yellow for a long time? Well, when you view yellow for a long time, yellow activates a green receptor, and yellow also activates a red receptor. As a consequence, these receptors adapt. So then when we activate the two by a white light, we activate this one and this one by a white light. This one gets a normal response to white. This one does not get activated by the yellow part of white, and so as a consequence gets less active. And the overall perception then is that there's a blue center here. So when we look at opponents, the opponents, double opponent cells of yellow and blue have one opponent cell. If, on the other hand, we looked at this purple, its opponent should be green. So in fact, if we pick any of these colors and just go to the opposite side of this color wheel, we'll find its opponent. What are some of the other properties of these cells, these double opponent cells? One of the things is what color will maximally activate these cells. Okay. Here we have a double opponent cell in your cortex firing, not your normal ganglion cell, but one inside your cortex now. And what was its response to different colors of light? Well, when we shine yellow light on this, okay, it decreases its response. When we shine blue light at this, we find an increase in activity in this cell. So as a consequence, that we have a double opponent cell that's sensitive to blue in the center and inhibited by yellow in the surround. If we have this type of cell, what will maximally activate this double opponent cell? Well, if we try blue in the surround, we cancel it, okay? What happens in this center is the opposite of what happens in the surround. In contrast, when we put yellow light in the center, we have a huge activity in this ganglion cell, in this double opponent cell. So blue light, a blue center against the yellow surround stands out very well. And you'll find that these complementary colors are colors that stand out from the background. These are colors that these double opponent cells are particularly sensitive to. What happens when we have light that fills, of a single shade that fills these two? If we shine blue light over this double opponent cell, nothing happens. There's no change in activity. If we shine yellow light over this double opponent cell, again, no change in activity. In fact, any color over both the center and the surround produces no change in activity. So these cells have a color constancy. They are insensitive to a color that fills both the center and the surround. And as a consequence, an apple looks red by the noonday sun or by the evening sunlight. Okay, there's a huge change in color that's hitting that apple when it's sunset. Okay, lots of reds involved, but that apple still looks red. It maintains its constancy. The same thing doesn't happen in a camera. Cameras, if they're not set up properly, have a yellow hue when you take in the indoor picture, and again, if they're not set properly, will have a blue hue when you take an outdoor picture. They have no color constancy. So in summary then, the eye does three things. It focuses the image clearly on the retina, if those lenses work properly. It then detects colors and different intensities of white and black, gray, and then compresses this information, taking a lot of information from the fovea and less from the periphery, and sends it down the small optic nerve. Its compression occurs in two ways. First of all, as I mentioned, only a detailed picture is sent from this small 1% of the retina, and only that information from the edges is sent, okay? Not that from the surround. And as a consequence, only changes in only activity of a few receptors can allow you to see clearly. Now, this course is called Transformations for Action and Perception. And we're looking at different ways information from the retina, for example, gets transformed into the activity of the ganglion cells. And at first sight, it seems like this is what first appears on the retina is much better than what comes out of the retina. And how this, what we think is a poor image, then gets transformed by the cortex in what you see every day as a clear and vivid picture will be the subject of the next few lectures. Thank you.